ATP, or adenosine triphosphate, is a primary energy currency in a cell. It produced and regenerated through multiple metabolic pathways, each tailored to manage energy demands during rest and activity. In this presentation, I'll explore how ATP is synthesized and recycled using glycolysis, the TCA cycle, oxidative phosphorylation, and anabolic pathway like nucleotide and fatty acid synthesis. This diagram shows the steps of glycolysis starting from glucose. Enzymes like hexokinase and phosphofructokinase consume ATP to phosphorylate glucose and fructose 6-phosphate. Eventually, each glucose splits into 2 3-carbon molecules and glyceraldehyde 3-phosphate. These are oxidized to form pyruvate, yielding a net gain of 2 ATP and 2 NADH per glucose molecule. This is the cell's first way of generating ATP quickly without needing oxygen. Pyruvate is then converted into acetyl-CoA by the pyruvate dehydrogenase complex. This is a key regulatory step, linking glycolysis to the TCA level. It also generates NADH, which will be used later in oxidative phosphorylation. Acetyl-CoA serves as a substrate for the citric acid cycle and for other biosynthetic reactions. In the TCA cycle, acetyl-CoA combines with oxyl acetate to form citrate. Through a series of reactions, this cycle produces 3 NADH, 1 FADH2, and 1 GTP per acetyl group. These electron carriers are the cell's primary means of feeding electrons into the electron transport chain, which is where most ATP is generated. This diagram represents the ETC embedded in the inner mitochondrial membrane. NADH donates electrons to complex 1, while FADH2 enters through complex 2. Electrons transfer through coenzyme Q to complex 3, then to cytochrome C, and finally to complex 4, where oxygen is introduced to water. An electron move protons H+ are pumped into the inner membrane space, setting up the gradient needed for ATP synthesis. Here we zoom into ATP synthase. The enzyme works like a rotary motor, with conformational changes in its beta subunits, driving the synthesis of ATP from ADP and inorganic phosphate. The proton motive force drives the rotation of the gamma subunit, cycling through loose L, tight T, and open O states. Each full rotation synthesis 3 ATP. Here we see how amino acids like alanine, aspartate, and glutamate are converted into TCA cycle intermediates. For example, alanine is transminated into pyruvate, aspartate, and into oxyl acetate, and glutamate into ketoglutarate. These reactions allow amino acid to feed into ATP-producing pathways during fasting or exercise and catalyzed by enzymes like alanine transaminase and glutamate dehydrogenase. This slide outlines cholesterol synthesis from aspirin units, dimethylol triphosphate or DMAPP, and isopentenyl triphosphate or IPP, condensed into larger molecules like farnesyl pyrophosphate, which then forms squalene. Squalene undergoes oxygenation and ring closure to form lanosterol, and finally, through 19 steps, cholesterol, NADPH, and ATP are required throughout, linking this anabolic pathway to energy demand. This shows the conversion of methionine to succinyl-CoA and TCA intermediate, though through intermediates like S-adenosylmethionine or SAM and cysteine, methionine is broken down, eventually forming propanol CoA, then succinyl-CoA. This path integrates amino acid metabolism into energy production, particularly when glucose is low. Together, these pathways form a network of ATP production and regeneration, glycolysis, the TCA cycle, and oxidative phosphorylation yield ATP, while amino acid, fatty acids, and even cholesterol precursors contribute to or consume ATP on metabolic needs. Understanding these pathways help explain how our cells stay energized, adaptable, and resilient. And look at the last slide to see a full picture of each and every single step of the metabolic poster. Thank you.